Now let's talk about some of the biological activity in lipids. We've discussed a lot about the different types of lipids, and in particular our sphingolipids, our membrane lipids, and triacylglycerides and glycerophospholipids. Now, all of these types of membrane lipids can be put together and degraded. Now, I focus on a second just to talk about how that happens. Now, because we have to continually degrade and replace membrane lipids, there are special enzymes or proteins that help us chop apart and put together different lipids. These are called phospholipases. Lipases, that is, lip is for the lipid. Ase means to cut. So a lipase is a enzyme that cuts our lipids. A phospholipase, particularly, especially, cuts lipids that are a phospholipid. They have to have a phosphate group there. Now, these phospholipases, there are different types, and they only act on certain areas of a lipid. They are distinguished by their lettering and also numbering. So there can be phospholipase A, and it goes through to D. And each will only cleave a very specific bond on a lipid. So you can see here, for example, this is a glycerophospholipid. We have a glycerol backbone and a phosphate group. And you can see these phospholipases can cleave at different points. This phospholipase A1 will cleave the fatty acid off of position 1 of a glycerol. Phospholipase A2 cleaves the fatty acid off of the second position of a glycerol backbone. And phospholipase C will cleave this whole phosphate and head group off. Now, what if you just wanted to detach the actual head group away from the phosphate? You would use phospholipase D, which just takes the head group portion off. Now, if you have dysfunction in this area in your types of phospholipases and actually moving of sugars in particular on your on your lipids. So, for example, this particular structure is a this is a glycerophospholipid that has a sugar head group or a ganglioside. And if you do not degrade these types of lipids correctly, you can have buildup of lipids in a lysosome. Lysosomes, if you remember, are the area of the cell where you break things down, quite frankly. And if you build up too much excess lipid in your liposome, you can have disorders that eventually can lead to bigger problems. So, for example, failure at each of these points, you can see these are different enzymes that have failed in their ability to break off a piece of a lipid, leads to different diseases. So, for example, this sphingomyelinase, which helps to break off the phosphocholine of a sphingomyelin, leads to Niemann-Pick disease.